a lot of what we do focuses around fish, and salmon specifically. And so to have such a renowned fisheries biologist speak to us today is really an honor. So it is, again, my privilege to introduce Dr. Daniel Schindler, who's going to present on his research on salmon rivers in western Alaska. Dr. Schindler is a professor in the School of Aquatic and Fisheries Sciences at the University of Washington. He is a principal investigator of the university's Alaska Salmon Program, which has studied the ecology of salmon and their watersheds in western Alaska since the 1940s. His research is focused on freshwater ecosystems and their watersheds, addressing questions ranging from understanding basic ecological and evolutionary, there it is, and evolutionary processes to the effects of climate change, watershed development, and fisheries on ecosystem dynamics and natural resources. He's a previous recipient of the Frank Rigler Award from the Society of Canadian Limnologists, the Carl R. Sullivan Fishery Conservation Award from the American Fisheries Society, and the G.E. Hutchinson Award from the Association for the Science of Limnology and Oceanography. He was elected to the Washington State Academy of Sciences in 2018 and recognized as a Fellow of the American Fisheries Society in 2021. A quick Google search on Google Scholar will reveal that his publications have been cited over 28,000 times. And finally, a fun fact, Daniel is also a world champion dog sledder. So, right? That's amazing. So, anyway, thank you. Please welcome Dr. Daniel Schindler.
All right, thanks, Glenn. And thanks for the invitation to speak with you today. It's a pleasure to be here, and thanks to Glenn and to Judy for the invitation. You guys are really into technology here, so I'll see if I can figure out how to use it all. So I gather most of the people, or a lot of you, are engineers and hydrologists and geomorphologists, so I'll try to give an overview of some of our work in western Alaska that's trying to understand how salmon and some of the animals they interact with respond to the geomorphic template and the changing geomorphic template of watersheds in Alaska. And this term I want to really dwell on is the thought of a mosaic of habitat, which is something that I think everyone thinks about when we consider what habitat looks like in aquatic systems. And really the emphasis I want to place is that this mosaic of habitat is not static. This is a set of conditions that varies spatially and temporally. So that mosaic is continuously shifting through time as climate and geomorphology interact, and then the biology responds to that changing mosaic. Thanks. So I guess we're not allowed laser pointers, but anyhow. So I'm going to talk about western Alaska here. Most of the work I'll talk about is in Bristol Bay down there in the southeast part of the Bering Sea, and then a little bit about the Kuskokwim and the Yukon. And these are places where salmon rule. In Bristol Bay, I'll talk about sockeye salmon, which in the last decade have reached levels that are higher than ever imagined in the past. The record return of sockeye salmon occurred 2 years ago, where 82 million fish returned, despite over 120 years of intense harvesting on it. In the Yukon and Kuskokwim, the dominant species are chum salmon and Chinook salmon, which in the last decade have really crashed. And the subsistence communities throughout the Cusco and the Yukon have really suffered because of declining salmon populations. So a lot of the work that I'll talk about here is really motivated by trying to understand why are fish changing in abundance, how does habitat interact with climate conditions to affect both the numbers of fish returning to systems, the reliability of those fish, and of course what this means in terms of what we call ecosystem services and fisheries production to subsistence and commercial communities. So here again, a term here's a concept that of course geomorphologists have thought about for a long time, is the idea that habitat is nested and structured hierarchically. So in the case of the fish I'm talking about, up there in the top left, these fish are spending about at least half of their lives in the marine system in the Bering and the Gulf of Alaska. And as they return to Bristol Bay, in the case of sockeye salmon, they separate out into 9 major rivers. So they're homing to their natal rivers. And then within each of those river systems, we have dozens, if not hundreds, of individual populations that are locally adapted in an evolutionary sense to local habitat features that they encounter for spawning and for juvenile rearing. And this is the hierarchy that we want to talk about, and one of the messages I hope to impress upon you today is that there's relevant and important ecological variation expressed from the finest of those scales at individual populations all the way up to the North Pacific Ocean. So if we look at how many sockeye salmon have returned to Bristol Bay, like I said, the commercial fishing started in the late 1800s, basically after we trashed the Columbia and caught more fish than we probably should have. Those same boats and some of those industries packed up and headed north to start harvesting sockeye salmon and put them into cans. So we're now 140 years later, and there's more fish coming back than ever before. Part of that has been because the habitat's intact. Part of that's been because the management has been successful, and it's also because the climate has turned to favorable conditions. But if we look at the last 60 years or so, if we just look at the number of fish coming back, back in the 60s and 70s, we got 20 to 30 million sockeye returning to rivers, and as you can see, the last decade, we're upwards around 60 million fish. This is without any propagation or any enhancement or any restoration of any type. But the more important message here is gleaned by looking at the colors. Each color here is the number of fish returning to individual rivers. And if you go back to the 60s, you can see that most of the fish were coming from a single river system, the Kvichak in the yellow there. And after the PDO shifted in the late 70s, we see this proliferation of all those colors, okay? So now in the last 50 years or so, there's been a much broader diversity of rivers that have contributed to production. So the production that has increased in the last 10 years was particularly pronounced in the Green and the Gray River there. That's the Wood River and the Nushagak River. And if you go back to the 60s, you see that those rivers had contributions that were so small they were hardly even monitored, okay? So if we were developing these watersheds in the 60s, it would have been really easy to emphasize that yellow river, the Kvichak, and say this is where we need to put all our habitat protections and our conservation efforts. And if we're going to develop something, we could look at some of these smaller rivers. Had we done that, had we compromised the habitat back in the 60s and 70s, there wouldn't have been this opportunity for that Wood River and the Nushagak River to build to the point that they are now, where they're upwards of about 30 million fish, about 10 times higher than they were in the 60s. So why is this? Some of this has to do with, we think, climate change. So in blue and orange there is the number of fish returning to the Wood and the Kvichak, the Wood and the Nushagak. And you can see that after the 70s, and particularly in the last decade, they've really taken off. In the black bars is one signal of climate change on the physical attributes of the system, and this is the spring ice breakup date, which correlates with summer water temperatures and zooplankton production in the lakes, etc. And you can see that since the 70s, and particularly in the last 15 years, ice breakup date is occurring earlier and earlier. And what that's doing is extending the growing season. There's more plankton in these lakes. The fish are leaving as bigger smolts. And in these two systems, for whatever reason, they seem to have been in the deep freeze up to about the 70s. Climate warming has opened up their productivity, and that's where a lot of the production has come online. So I mentioned earlier, it would have been easy to write these systems off 60, 70 years ago because they were not an important contributor in a proportional sense, but by staying in the game, they became the big producers that we've seen in the last 10, 15 years. And I just want to give you an image of what this looks like when we get really high runs. This is one of the lakes within the Wood River system. In 2018, had the record escapement of 8 million fish to a relatively small river. And the fishery took about 3/4 of them. And this is what's left when there's only 25% of the run left on the spawning grounds. It was insane. So this is in mid-September. This is a little earlier than when all the fish are spawning. And you can see there the small dots near the beach are fish that have set up to spawn, and those dark clouds of fish are fish that are queuing up, waiting to spawn. It turns out that somewhere on the order of 80 to 90% of them probably didn't spawn successful that year because there are just simply too many of them and not enough habitat. But the point is that one little lake here at the scale of Western Alaska produced this insane number of fish that year. So let's put that in the context of global production of sockeye salmon and ask, well, how important can an individual piece of habitat be to global production of salmon? So we'll start with roughly 400 million pounds of sockeye were caught in the world in 2018, and Bristol Bay accounted for about 57% of that. Bristol Bay is, on average, about half of the world's sockeye production. And then if we ask, well, how much of that was caught in the Nushagak fishing district? This is where I wish I had a pointer, but you can see sort of on the north side of Bristol Bay there's a fishing district called the Nushagak that catches fish returning to the Nushagak River and the Wood River. The Nushagak district alone was about a third of the global catch. If we then carve out how much was in the Wood River, so the Wood River is a minor tributary of the Nushagak, the Wood produced about 20% of the global catch of sockeye salmon in 2018. And then if we ask, well, where did the fish return to in 2018 within the Wood River? And this is where the escapement went. And Lake Beverly, if you count up from the bottom, is third lake up. And you can see on the west side, there's a little finger that pokes out. Most of the fish returned to that little arm of the lake, okay? So somewhere on the order of 4.5 million fish escaped, so that means swam through the fishery and went back to spawn in that one single little piece of habitat in that lake. And if we then ask, well, how much of the global production of sockeye came from that one lake, it's about 13% in 2018. Typically, it would be rounding error at the global scale for this one system. So it turns out Lake Beverly, if you add up all of the salmon habitat in Western Alaska, sockeye salmon habitat in Western Alaska, it's less than 1% of all the habitat that can produce sockeye. So 1% or less than 1% of Western Alaska sockeye salmon produce 13% of the global catch. And in fact, if you look at the global distribution of habitat, there's probably 10 times more habitat at the global scale. And like I said, some tiny, tiny fraction of that produced 13% of the global catch in that single year. So the point there is that, again, it's easy to write off small pieces of habitat simply because they don't have fish in them. But if you think about their production potential, every now and then little pieces of habitat go off and become disproportionately important to a broader aggregate. So when we think about how systems operate, you have to keep the texture, all those little pieces in play, because they will often become important at some point in the future if we haven't seen it already. And I know there's a lot of thought at this conference about habitat and monitoring. And if we assess how important is an individual piece of habitat, it's really important to keep this in mind, because short-term assessments of how important a piece of habitat might be will generally underestimate the potential of that system. So here's some data starting in 1946. On the left side is a little creek called Happy Creek that every year for the last 70 years now, we walk the whole stream several times of the year to count how many fish escaped in it. And back there in the 60s and 70s, it only had a couple thousand fish in it. And then for some reason that we still don't understand, the light switch went on, and that system now produces 20 to 25. It doesn't produce, it has 20 to 25,000 fish spawning in it every year. Production is about 3 to 4 times that. So again, it ground along at not much of anything, and then the light switch went on and it's become very productive. On the right is a similar creek, not very far away, same watershed, and that's what its production has done over this time period. It's marching to the very different drum, okay, even within the same watershed. So what we have here is that any given site is quite volatile in terms of its productivity for fish, and even within a single watershed, all the tributaries have very little coherence amongst them. So we call this response diversity. Even within an individual river system, individual populations respond in very different ways to the same regional climate forcing. And this is not because one population is harvested heavily and another isn't. It's something else about the habitat-climate interaction that we don't understand. And what does this mean for monitoring? This is work of Brooke Davis, who did her master's thesis on this. And we did this analysis because at the time, Pebble Mine was still is proposed, but at the time it was really heating up in terms of a proposed large-scale gold, copper, and mostly sulfur mine in Bristol Bay. And the people doing the risk assessments would go in, do 2 or 3 years of stream counts and say, these little streams that it's going to drain are 1 or 2% of the overall production. So our question was, all right, how wrong could they be by stating it's only 1 or 2% because they've only looked at the system for 3 years? So this is complicated, but these are 8 streams that we have long-term counts on. And what we've done here is ask, well, what proportion of the total watershed or of the stream production in these systems comes from any individual stream? And you see that it's quite variable. An individual stream can be as low as 1% in some years and 30 or 40% in another. And on the top there, top right, is the probability, according to different monitoring windows, of seeing something within the top 75% of productivity. And you can see that even with 4 or 5 years monitoring window, you have barely more than 50% chance of seeing the maximum productivity potential of that piece of habitat. And even after 10 years, you haven't observed the max, or even within 75% of the maximum productivity potential of that stream. So when we monitor these things, we've got to be in it for the long run if we're going to estimate how important could that piece of habitat be in the future. And in fact, these things rarely ever asymptote. The longer you watch, the more productive you're likely to see that system be. So one criticism or one thought, if you consider this, is what we're looking at so far is really the individual year-to-year buzz in the data. And maybe that over longer time scales, where we have bigger climate regime shifts, that what we're looking at is a sort of a regional climate signal that then individual streams have short-term buzz associated with it. So we asked this question of whether such response diversity in the populations could play out over centuries. We don't have data that goes back for centuries, but we can use the paleogeochemical records at the bottom of lakes that accumulate in their sediments to reconstruct how many salmon were there in the past. And this is a very complicated figure, but you don't need to know the details. Top left is the rig we use to drop a core and device into the sediments. We get a tube of sediments that we can then slice up into individual slices, and that represents chronology of history of that lake. And because salmon are coming back from the ocean with a distinct nitrogen isotope signature, we can analyze how heavy the nitrogen is in those sediments, which is proportional to how many fish spawned there in the past. And what I'm plotting here, a time series for 500 years for lakes that produce about 70% of Alaska's sockeye salmon. The green boxed lakes are reference lakes that have a waterfall and no access to salmon. You can see that there's very little variability in them, and their isotope values are fairly low. They're down around 2 or 3. All the other lakes have salmon in them. And pick pairs of lakes and ask yourself, how much coherence are there in the ups and downs? And you can find pairs of lakes. For instance, on the right-hand side, those are all lakes on Kodiak Island. You can see that they sort of go up and down roughly in concert with each other, but they're not coherent with what's going on in other parts of the landscape. And even within Bristol Bay, which is most of those other lakes, different lakes have different wiggles in the data. And this isn't measurement error. We've looked at this in various ways. This is individual rivers and individual lakes showing their own dynamics with frequencies that are sort of at the 1 to 200 year scale. And in fact, if you just do a simple principal components analysis on all those time series and ask, what's the coherent temporal signal in the data, there's only one principal component that's significant, and it's the blue one here. And most of what it's chasing is the decline in salmon escapement to those lakes seen in the last century. So remember, the fisheries came in around late 1800s, intercepted a lot of those fish. So the isotopes that were in those bodies of fish weren't deposited in lake sediments. They were put in cans and sold in Britain and Europe and places like that. So this actually confirms our method, which is that all those lakes show a decline in the last century. But aside from that signal, there's nothing coherent across Western Alaska in terms of how many fish came back to these lakes over the last 500 years. So response diversity, even in closely adjacent lakes and rivers, is highly variable. There's lots of response diversity, even at long time scales. So this brings up, questions about, well, how do we define what critical habitat is, both at small scales and at large scales? And we've tackled this question in a variety of ways. Some is using the long-term data, and some is using the isotopes that we can get out of the otoliths in fish returning to lakes. These watersheds are remote enough that you can't really sample them, but you can ask for fish returning to them, take their otolith out, which is a little ear bone in their head that is laid down chronologically like the rings on a tree, and the water chemistry of the water the fish is swimming around in at any one time is incorporated, the trace elements are incorporated into the otolith. So this is work by the late Sean Brennan, who died unexpectedly a couple years ago. But what he did was take the strontium isotopes and produce what we call an isoscape, which is just the variation in the isotope ratios across, in this case, the Kuskokwim River, and then take fish that are caught coming out of the ocean, pull out their otoliths, and ask where was that fish born. So in the middle panel there is as a probability distribution of the most likely places that individual fish had been born in. And then you can take a bunch of samples and aggregate up to what the watershed produced in that year. So that's a heat map of where the Chinook salmon production was in the Kuskokwim in this year. And we've done this for a couple of rivers now and asked, well, how consistent is it from year to year? And is this a way to identify where the critical pieces of habitat are in these really vast and difficult to sample watersheds? So I'll start with the Nushagak River. Here's a sockeye fishery. It intercepts some Chinook salmon, such as that one shown there. And the question is, where did the fish come from that were caught in that year? So for 2011, this is what the heat map looks like. And the redder the color, the more fish were inferred to come from that tributary. And you can see in this system in 2011, most of the fish came out of two pieces of the watershed, that tributary in the north central part of the watershed, and then a little bit from the northwest piece of the watershed. And in fact, most of that watershed didn't produce much anything that year. And I wish I had a pointer, but on the east side, on the right side of the screen there's a little forked watershed about halfway up. That's where the Pebble Mine would have sat, right in the headwaters of that. So if you were to assess, what's the potential impact to fish in the Nushagak River from, for instance, spillage from this mine, you would look at a map like this and say, not a lot of fish on the eastern part of the watershed, probably good to go. Well, we did this another year, and this is what it looks like in 2014. That central northern tributary didn't produce much of anything that year. Most of the production shifted to the east, and the Mulchatna River then became the important tributary. And because this took so much work, I'm going to go back and forth and show it off a little bit. So what you can see is that you have a big watershed, or by Columbia scale, this is a small watershed, but the production is coming out of different parts of the watershed in different years. So we also did this for the Yukon and the Kuskokwim, and these are literally off the press a couple days ago from a graduate student who's picked up the work that Sean Brennan started, and we'll show you the Yukon River now. Again, it's a river that's 2,000 kilometers long, difficult to sample throughout, but the same trick where we sample fish coming into the river if they move out of the ocean. This is for Chinook salmon again, and then ask where were they born. So there's Ben who's done this work. The star is where the fish were caught, and that's our production map for 2017 for the Yukon River. White colors didn't produce many fish, or that we didn't detect many fish from that part of the watershed. Red is where the fish were coming from. So in 2017, it looks like the Yukon produced fish from throughout the watershed. It was a relatively poor return that year, but they seem to come from everywhere in that system. And now if we go to 2019, it looks like most of the fish in the system came from Canada. The Canadian border there is about where it turns red. And then in 2021, you can see that it's somewhere between those two. So we'll just step backwards through these if I can do that. Again, you have this mosaic of production, and the mosaic is continuously changing. Can you guys see this thing changing? Okay, this isn't because you drank too much coffee. This is what we infer is going on in this system in terms of where the fish are coming from. So then you ask yourself, well, what is the critical habitat in the Yukon River or the Nushagak River? And it looks like almost all of it is critical, but it's not critical in any individual year. So if you want that system to function as an ecosystem, you have to keep as much of that habitat in the game as you can. And this applies both to conservation of habitat, but also in the case of restoration, restoration needs to at least plan at the scale of the whole river, okay? And it's not to say that individual small pieces don't matter, because they clearly do, like in that case of the Beverly Lake that I showed you earlier. Little pieces can light up, but if you're interested in the system and how stable it is and how productive it is, you have to assemble all those pieces into one aggregate and think of it at the scale of the whole river system. Okay, moving forward. So what about really small scales? So it's really easy to say, all right, everyone has to think about stuff at the biggest scale possible. Well, I just want to give you two more examples of why habitat variation at really small scales is also critical to the ecology of these fish. And this is work of Johnny Armstrong, who's now a professor at Oregon State University. And for his PhD thesis, he looked at juvenile coho salmon, the striped fish there, who's chasing down a terrified little sockeye salmon. And they spawn and rear in these small streams in western Alaska. And it turns out that those coho salmon get big by eating the eggs in particular of sockeye salmon that are spawning in the same streams. So there in the top left is a juvenile coho salmon, a couple inches long. It's got a bulging belly. This is the way I felt last night after dinner. And it is full of sockeye eggs, okay? And there's the sockeye down in the bottom. This sort of ugly graph here is a map where the top of the system is in the top left, and it trickles downstream. That's about 2 kilometers of stream down to the bottom where it exits into a lake. The color is the water temperature. So it's warm at the surface. It's a bunch of beaver complexes, and at the bottom it has spring-fed groundwater that's cooling down the water. The size of the bubble is how many sockeye salmon spawn in the system in that year. And you can see that the sockeye are piled into the lower part of the stream where the water's cold, there's lots of oxygen. It's probably more stable flows. Well, what was interesting is in Johnny's first year in his grad program, he did snorkel surveys up and down this system. As you'll see in a second, it's a really small system. And he found those bloated coho salmon way up there in the red. And the question is, well, how are they getting these sockeye eggs? Because the sockeye eggs are a mile away. So the thought was, well, maybe the sockeye are coming up at night when we're not there because we're scared of bears or something. Or maybe those little coho, which are literally the size of your finger, are going down to get sockeye eggs. So what Johnny did was, so there's an overview of the stream, roughly the different regimes of water temperature. We put some antennas up where the yellow dots are, and then we put pit tags in fish at the top. These are little coho, put them in the top, turned on those antennas, and then listened for them, okay? And there was a couple options of what could have happened. Those little fish could have gone down, fed on eggs, and then swam their way back up, or the sockeye could have moved up and spawned at night. We don't think that was what was going on, but we had a hard time believing these little coho would navigate that little stream there for a mile, basically, each way to get eggs. This is our best behaved coho, but a lot of them look like this, okay? So section 3 is that warm site at the top, and that's the residence of that fish in that warm section of stream. So we tagged it, and 3 days later, it migrated down, hit 2 antennas, went down into the cold site, spent 3 hours, turned around, swam back upstream that same night. And then it sat there for 3 days, and then it went down, spent a couple hours in that cold part of the stream, presumably binging on sockeye eggs, swam back up to the warm site, spent 4 days there. It did this all summer. And like I said, this was our best behaved individual, but a lot of them did this as well. So what's going on here? Water temperature exerts control over the metabolism of fishes. And if they eat a big belly of eggs and sit in cold water, it takes them about a week to digest them. If those fish move to 14-degree water, they can basically move and process that food in about 3 days. So what we're seeing here is they're migrating up to sit in warm water and metabolize and digest that food, empty their guts, and go down for another binge. Okay, so what's going on here is we don't know how they're doing this, but these little fish the size of your finger are figuring out where the warm water is, and they're exploiting that heterogeneity at the scale of hundreds to maybe 2 kilometers of stream. So we were excited. It's like, wow, this is really cool. So in A there is 2017, the year that Johnny discovered this. 2008 is the year we first put in those pit tag readers. The results look too good to be true. So we're like, all right, let's do this one more year. And in red in each panel is the upper stream temperature. The blue is the lower stream temperature. So you can see in 2008, we got big contrasts in how warm the temperature was at the top versus the bottom. And we saw the fish exploiting the thermal heterogeneity. 2009 rolled around, we did the same thing, and it rained every day that summer. And what you can see is that the temperature variation went away. And the bottom of the stream and the top of the stream had exactly the same cold water temperature. Basically, there was too much water flowing through the system, and this upper parts simply didn't warm up. Johnny was distraught. I said, don't worry, we'll do it one more year and it'll all be fine. So 2010, we did it again. Same thing, lousy year. We got wet, and the river was at basically just below flood stage all summer long. No temperature variation. And luckily, we did it one final year in 2011. It was a dry year, and once again, we got that temperature variation, and the fish were going back and forth once again. So after 5 years of collecting this data, we thought, all right, we have this awesome data set, because what we're going to show is that in years where there's thermal variation, the fish exploit it, they're going to grow faster. And in wet years, when there's cold water everywhere, they won't be able to capitalize on this high energy subsidy of sockeye eggs, and they'll grow slower. Well, we analyzed the data, no difference in growth rate among those 5 years. So we're scratching our heads thinking, what the heck is going on here? So much for that theory. And we dug into it a little bit more, and this is a really fancy graph, I guess, but let me step you through it. Each vertical line is the thalweg of those streams. So look in the bottom left, 2007. The temperature of the trunk of those little drawings is the stream temperature. So in 2007, you can see the top of the stream is warm, the bottom of the stream is cold, and the blue dots at the bottom show you where the sockeye are spawning. So 2007, 2008, there is warm water at the top, cold water at the bottom. 2009 and 10, it's cold throughout. 2011, it's warm at the top, cold at the bottom. And this is all a response to how much precipitation we're getting. In all years, the sockeye are packed into the bottom of the stream where they're spawning, where upwelling's coming out of the groundwater. In the top is where the little coho were. So if we look at the first 2 years, they're up at the top of the system, sitting in warm water in the thalweg of that stream. And then the 2 wet years, the dot is the size of the dot is the number of coho in that stream section. The color of the dot is the water temperature they're sitting in. And what you can see is that in every year of this study, coho are sitting in warm water. So how is that? In dry years, the water in the stream is down in the thalweg, and you get longitudinal temperature variation. And this is, maybe the geomorphologists are rolling their eyes now, but in the wet year, the river floods up, spills out, and there's now lateral connections to off-channel habitat that warm up. We don't know how the fish are figuring this out, but when it's dry, they're going up and down the stream to find warm water. When it's wet, they're going laterally to find warm water. So the point is that the thermal temperature, the temperature requirements for these fish are met independent of what the hydrology is. When it's a wet year, they find warm water. When it's a dry year, they find warm water. And this is just an expression of the geomorphic complexity in this system. And the fish have evolved mechanisms that we don't understand, we probably never will, to seek out those resources and make use of them. I want to, one final example of why the geomorphic variation and the hydrologic variation across the system matters to understanding how these systems function is to look at some of the other species that eat salmon when they return to spawn in lakes and rivers. So there's a nice Arctic char that's going to binge just like those coho did on sockeye eggs. And it turns out that the opportunity that the consumers have in these watersheds, so these are things, everything from char and rainbow trout and bears and birds, all these things eat sockeye tissues and sockeye eggs. It turns out the opportunity for them ultimately depends on the geomorphic conditions within the watershed. And I'll motivate this by showing you a bear eating a salmon, a little sockeye salmon, and that would happen a little quickly, but I'm going to go back one more time, I think. Yeah, so just watch the bear is going to catch a salmon. It's like a 400-pound bear catching a 5-pound salmon, and look how much it eats. So it grabs it, snacks a little bit, eats the brain basically. So these bears typically will eat the brain of the males and the bellies of the females and eat their eggs. They don't eat the whole fish, okay? And the question is, why are they eating only little tiny pieces of these fish? And again, come back to the buffet dinner last night. Basically, there's so much food here that they don't want to fill up on a bunch of junk food. So what they do instead is they really high grade for the most nutritious tissues. And if it's a female salmon, the most nutritious tissues are eggs. If it's a male salmon, it typically is the eyeballs and the brain. So what this says, and this fish biologists haven't got this for some reason, but if what you think about what's going on here is the bears are not constrained by how much food is available to them. What they're constrained by, just like those little coho salmon, is the time they need to capitalize on it, okay? So what they're doing is high grading for the highest quality tissues, just like the food buffet last night. I skipped all the salad and all the rest and went for the salmon. The bears are doing the same thing. There's fish everywhere. They could eat 3 or 4 or 5, maybe 10 salmon and be full, but they don't do that. They kill hundreds of salmon and eat the highest quality tissues because they're time limited, not resource limited. So it turns out that whether it's a little coho or whether it's a bear, time is a really limiting resource because these fish, the salmon, are only present in the watersheds for a couple months of the year. So how does geomorphology then regulate time available? So this is the Wood River system. This is work by Peter Lisi for his PhD thesis, and each dot is a different site that has fish spawning in it. The color of the dot is the average summer water temperature that ranges from 5 to 13 degrees. Top right there is the spawn timing of salmon of a local population in that stream as a function of water temperature. And when the water temperature is cold, fish spawn in it earlier, presumably because it takes longer for the eggs to develop. Warm sites, fish delay and don't spawn in those warmest sites until September and even, in fact, into October. And the reason the cold sites are cold is because they're in certain areas where snow is melting, filling up local aquifers, and then dribbling out into the stream over the course of the summer and keeping them cool. So there's a climate change piece of this that you can ask me about later. I don't have time today to talk about it. But the question is, does this variation in spawn timing by salmon translate into opportunity for bears? And of course, the answer is, well, if the bears can move from site to site, they can extend how long they have access to salmon for. And we just did a simple study where we put game cameras up on streams and asked how many bears are here. So this is one site that's cold. It's about 5 degrees Celsius for the summer. Put a camera up in the middle of June. You don't see any bears until the middle of July. And literally within a day of the salmon entering the stream to spawn, you start seeing bears. And the camera buzzes away for 3 weeks and takes lots of photos of bears. And then about the time the fish are done, which is about 3 weeks later, you don't see any bears there for the rest of the season. So that's one site that's cold, and if I overlap, this I guess is 5 different sites, what we see is bears are all packed onto small cold streams early in the season, and then they move to other parts of the watershed, which may only be 1 or 2 or 10 kilometers away, and feed on salmon at warm water sites later in the season. And we survey gulls. Gulls are scavengers. They're doing the same thing. They're all on the cold sites early in the season, moving to warm sites later in the season. And of course, local variation in hydrology, which is determined by geomorphic conditions, sets up this evolutionary opportunity for salmon, and that's what the consumers are capitalizing on. So that's just a time frame of when fish are available across a single watershed, and bears and gulls and rainbow trout can start feeding on salmon eggs, because they can't capture these things in deep water. They have to wait till they're moving to spawning areas where they get distracted and in shallow water where they're accessible or they die. But the consumers can start eating salmon in mid-July, and as they wander their way across the landscape, they sort of track this wave of resources that is tracking the hydrology and the temperature regime across the watershed. So instead of feeding on salmon for about a month of the year, they get to feed on salmon for about 3 months of the year. And that's probably why bears are so big and there's so many birds that are scavengers in Alaska. It's not because there's lots of salmon as much as there's a ton of geomorphic variation in the watersheds that provide this opportunity. So if we want to restore sites or protect sites to provide opportunity for wildlife, it's easy to focus on the big sites that produce lots of salmon. So those are the beaches on lakes and the rivers shown as the green and the red dots. And on the y-axis is the population abundance. Those are the big sites that produce fish. But in the blue dots are the small streams. So if you think about this problem as a time-limiting problem rather than a resource-limiting problem, you have to protect and restore all those little tiny streams, because that's where half of the opportunity for foraging opportunities by bears and birds are. So the small pieces really matter to the predator-prey system as well. So just to wrap up here, couple general messages, and again, some of these messages are old news to many of you who've studied the characteristics of fish habitat, but habitat variation is ultimately what stabilizes production to rivers, okay? It's not that there's just one good reach somewhere and it's gonna keep cranking fish out. It's the variation across the watershed that seems to stabilize production as certain parts of the watershed light up in some years and other parts light up in others. And this complexity is expressed all the way from coarse spatial scales to really fine spatial scales, and at long temporal scales all the way down to fine temporal scales. You know, this complexity is what produces the options for these animals to make a go of it in these watersheds. And if we think about climate change, you know, what can we do about climate change? It's going to be a long time until we dial back emissions and change the trajectory of the climate system. But what we can do is either protect or restore the habitat that provides the options for these animals to figure out how to make a go of it. And of course, the last piece is that infrastructure can mess some of this, particularly the fine scale stuff up. Alaska likes to think that it's going to do everything differently than the rest of the lower 48. They're making the same mistakes that we've made down here. Here's a road put in on the floodplain of the Chena River, I think this is. Yeah, Chena River. And of course, you can see the road is probably cutting off those off-channel habitats. There may be culverts in there that allow the fish to swim back and forth, but you know, the dynamics of that river system from a geomorphic standpoint have been taken away. And when we think about doing environmental impact assessments on things like the Pebble Mine, you know, it's easy to think about the direct footprint of the mine, but you have to think about the build out of the infrastructure and what that might mean to the whole watershed in terms of providing this heterogeneity in habitat conditions. And of course, down here, you know, a lot of you are struggling with how do we restore places like the Columbia to get some of that texture back in the system. And as you know, it's expensive, and in some cases, probably not possible. In places like Bristol Bay, most of those options are still on the table, and we got to protect that complexity before it's gone, and we're stuck trying to rehabilitate it, because as you know, it's very difficult to get back. So very briefly, the key messages, or this response diversity, is something we need to think about when assessing how a system is functioning. This heterogeneity is expressed across huge ranges of spatial and temporal scales. Third is, yeah, we'd like to do restoration at the big scale, and that's where the planning should occur and the coordination should occur. But even restoring small pieces of habitat can matter, because those small pieces of habitat often light up and become disproportionately important. And when we think about restoration and conservation, we have this history of making maps and planning around maps. But I would argue we should be making movies. Movies talk about the mosaic of habitat and how that mosaic changes through time. And that's what we should be planning around in terms of restoring processes and protecting the right habitat. And I just want to finish by showing some fish, and I'm happy to take any questions. Thank you. One question.
And I've got it. That's fantastic. Thank you. Very interesting talk. So a lot of the work that we do in the Columbia Basin, when we're looking at fish populations, one of the things we hear all the time is it's a bad fish year, it was because of ocean conditions. And we do this restoration work, it's like, yeah, well, but there's ocean conditions. And I'm curious what your thoughts are on the populations up in Alaska and how much ocean conditions are affecting these populations.
Yeah, ocean conditions are huge. That's probably why the sockeye are really booming in parts of Bristol Bay, is that both the watershed is productive and it seems to be increasing, but those smolts are going to an ocean that's really profitable. In the case of the decline in chum salmon, there's debate about the Chinook salmon, but it looks like the chum are suffering because of the warm blobs that developed, just didn't produce the right food web for chum. So there's no question that ocean conditions are a big part of the story, a huge part of the story. But those fish have to come back to somewhere, and they're not coming back to watersheds and just spreading out uniformly. The individual sites and the individual locations and situations in the watershed also matter and basically set the potential for those fish to do something when they hit the ocean. So it's really two filters that are at play. What we have control over is what's going on in the watersheds, at least to some degree. We have control over fisheries. We have no control over what's going on in the ocean. So that's the game we have to play.